Welcome to the 12th talk in the webinar series on genetics and evolution being organized by the Journal of Genetics. For this talk, I'm very happy to welcome Professor Amanda Bretman. She works at the School of Biology, University of Leeds. She obtained her PhD from the University of Leeds, working on sexual selection in field crickets. She then went on to do postdoctoral research at University of Exeter and University of East Anglia. And during this time, she did studies on the seminal proteins using RNAI and molecular mechanisms and fitness consequences of male responses to their rivals using Drosophila system. Professor Bretman has done pioneering work on plasticity in male reproductive behavior in response to changes in socio-sexual environment. She has used very successfully the Drosophila melanogaster system to do very elegant studies exploring how males perceive and respond to rivals in their environment and how this affects the number of progeny they produce and how long they live. She has won many awards, including the University of Exeter Merit Award for Excellence in Research. Today, she'll be talking to us about No Fly is an Island, how Drosophila respond to socio-sexual environments. Professor Bretman.
Thank you so much. Thank you for that introduction. That was absolutely lovely. And it's really lovely to be able to speak to you today about our work and also to show off my really excellent Photoshop skills. As you can see here, I'm really very good at that. So I'm going to talk to you today about fly social environments, and particularly how males are responding to their social environments in terms of reproduction. But I'm just going to very quickly say that this is the work of lots of people, and in particular, I want to thank the people I worked with when I was at the University of East Anglia, so Professor Tracy Chapman and also Dr. Claudia Fricker, and also my group at Leeds. So lots of the work in here is by Dr. James Rouse, and also Tom Leach and Lauren McDowell and my colleague Elizabeth Duncan as well. So I'm sure you are very aware that social environments are extremely important, and this is because they can change very rapidly and have profound effects on fitness and in very different types of traits. I'm sure you have noticed at the moment there's a little thing going on globally that is affected by our social interactions. And obviously, I'm talking to you from my spare bedroom and avoiding social interactions right now. But we can also think of social environments in terms of how they alter resource competition, disease, aging profiles, stress responses. They're involved in the microbiome, in cognition, in predation risk, and particularly in reproductive traits that I'm going to talk to you about today. And hopefully by at least the end of my talk, you will be very convinced that flies are interested in their social environment. But it takes some people a long time to accept that this is the case. But lots of people in lots of different labs are using fruit flies to understand social effects and to understand the effects they have on things like social learning in flies. So flies can learn about where there are parasitoids in the environment and when it's not good to lay eggs, for example. And it affects things like how flies sleep and how cancer progresses in flies as well. So although we think of them as very simple organisms, when I've heard somebody say they don't really have a social environment, they really are attuned to their social environment and it has really big effects for them. So I'm going to focus today, as I said, on the reproductive effects. And in particular, this problem that males have when females mate multiply. It means that they have competition for mating both before and after they mate. And obviously they don't have infinite resources to spend on this mating. And so there are trade-offs that males have to make between current and future investments and how they spend their reproductive resources. And so there's been this prediction for quite a while. So Jeff Parker devised the theory that this is based on, that males should be able to assess their social environment and use that assessment to invest in reproduction accordingly. So it might be that males are in this situation where they have lots of opportunity to mate, or they might be in this situation where they have lots of competition. And so they should be able to assess that at a particular mating and assess how much of their resources they should invest in that mating. And if we look around the natural world, we can see that this does happen in different ways. So these are just examples of males that respond to often either mating status of females or the presence of other males in their environment, and they change things about their mating strategy. So they can change their copulation duration, they can change their courtship efforts, or they can change their mate guarding. So there's sort of very clear behavioural responses. And they can also change their ejaculate. So the amount of either the number of sperm that they package or their seminal proteins that they package into an ejaculate. And that can go alongside these behavioural responses as well. So you can see that these plastic responses are very widespread. And that's sort of about as far as we go in many of these species. But I am going to focus on Drosophila melanogaster, and a real sort of accidental finding that I had when we were doing another experiment, and I noticed that males that had been kept on their own before mating mated for less long than males that were kept with other males. So this is the basic experiment that essentially 10 years of work is based on. And I'll show you a version of this experiment in lots of different manipulations. But basically we have males that have been kept on their own and males that have been kept with another male. And then we allow them to mate, and they mate on their own. And those that have been exposed to a rival will mate for longer. And you can see that it's in the region of a couple of minutes longer within a mating of between maybe 15 or 20 minutes. So we noticed this behaviour, and because we're using melanogaster, we can really start to unpick what's happening. So why males are doing this and how they're actually able to do this. And so in this talk, I'm going to sort of fill in these black boxes. So to think about the outcomes for males in terms of their fitness, but also their inputs in terms of their sensory inputs, for example, and within the slide, the mechanisms that enable males to be able to do this. So in terms of fitness, I'm now going to do a terrible injustice to a lot of work from ourselves and other labs as well, to basically tell you very quickly about the fitness outcomes for males. So we know, as I said, after exposure to a rival, males mate for longer. And this change in duration is largely under male control. And individual males can track their social environment. So you can switch males from being on their own to being with a rival, and they will change their mating duration in accordance with their social environment. And the effect that this has is that males that are responding to rivals have increased fitness, at least in the short term. And fitness in terms of, we've measured this both when they are the only male to mate with a female, so in terms of how many eggs that female subsequently lays and how many of those survive to adulthood. In terms of preventing the female from remating, so males that are responding to rivals can prevent the female from remating for longer. And when they're in competition, they have a greater paternity share as well. So at least over the short term, 1 to 3 matings, males that responded to rivals do better. And this change in behaviour is also associated with a change in their ejaculate. So they increase transfer of seminal proteins and they increase transfer of sperm and higher quality sperm as well. But there are costs to this. Obviously, if there weren't costs, then there would be no need for males to be plastic. But males are kept with rivals, age faster, so become progressively less good at mating as they get older, and they die sooner. So there seem to be costs to actually responding to rivals. So we can fill in some of that black box in the outcomes. So this behavioural change is flexible and it tracks the environment, and there are fitness benefits, but also costs to this response. Now, one of the key things that was predicted from the theory was that males should be able to adjust their investment depending on number of other males that were around. Now, we haven't actually found that flies can do this or do this. So they seem insensitive to the number of other males. Just having one rival is enough to produce a response. But what they do seem really sensitive to is time, so exposure time to other males. So first of all, we assessed how quickly the response appears. And so we kept males with a rival male for different lengths of time, and we found this linear response. So the longer a male is exposed to another male, then the longer his subsequent mating duration is. And then we can also ask how long the response is maintained. So when the rival is removed, how long does that focal fly keep maintaining that increase in mating duration? And we found that males maintain this response for around 15 to 18 hours after that rival has been removed. So they seem to remember that they have been with a rival. And these two properties interact. So exposure time affects the length of that memory of a response. So if we expose males for 36 hours, then they will keep responding after that rival male has been removed for 12 hours, but at 24 hours after removal, the response has declined. Whereas if we expose a male just for 24 hours, they do respond to that rival, so they make the response if we immediately mate them, but they very quickly lose that response. So at 12 hours here, they've lost the response to the rival male. So we think that for fruit flies, this property of time that they're spending with a rival male is giving them an idea of risk of competition. So we think that fruit flies are living in quite dynamic social environments. At times they are in very high density, maybe around a food source or an oviposition site, which is really the same thing. So around a rotting fruit, for example, they could be at very high density. But maybe they're also spending a lot of time on their own when they're flying between oviposition sites, for example. And so we think that flies are using this timing to be able to sort of work out whether or not it's worth responding to a rival. Is that rival around for long enough to actually be a competitive problem, or is that rival very transient in the environment and so not something to worry about? So in terms of inputs, we know that time with the rival is very important. And so we also asked what sensory cues are needed in order for males to respond. And so we addressed this question by using multiple techniques to interfere with the fly's senses both genetic and physical manipulations. So we altered either the rival being able to produce song or the focal male being able to hear song. So we used vestigial wing mutant rival males, so they have very crumpled wings and they can't sing. Or we used wild type males where we removed the wings of the rival, again, they can't sing. Or we used focal males, which are carrying this inactive mutant, which are hearing defective. We investigated odor cues by, again, trying to either remove an odor cue. So we used focal flies that were mutants for OR83B, or it's also known as ORCO. So this is an odorant binding protein that is an important co-receptor for a lot of the odors that flies can smell. We removed part of the antennae, which again is really important for flies' ability to smell. Or we tried to trick males into thinking that there was a rival around by supplying them with the smell of other males. So we extracted their cuticular hydrocarbons from other males, or we added a particular pheromone, the CVA pheromone, which has been associated with aggression in males and also aggregation. And actually, when we started this experiment, we thought that we knew the answer, and we thought that it would all be about CVA, and we were wrong. And this whole set of experiments turned out to be a lot bigger than we anticipated. We addressed visual cues, either using focal males carrying a white mutation, so they're vision defective, or we used wild type males held in the dark. And we investigated touch cues by having males separated by netting so that they couldn't touch each other, but they could receive all the other cues. So we investigated this by removing each cue individually, and in every case, males could still respond. So there was no single cue that removed this response to the rival. And so we had to basically do every combination of sensory cue removal so that we could work out what was actually necessary. And we came out with this really complicated picture where males are using any 2 of 3 inputs from sound, smell, or touch. So vision didn't seem to be important at all, but any 2 of these were required in order for males to respond. And we thought, well, one, it was very complicated in terms of our experimental design. It really turned out to be a lot more work than anticipated. But 2, we were wondering why it's so complicated and why there seems to be some redundancy in terms of the pathways that these sensory cues are taking. Okay, so we know something about this combination of sensory cues that males need. But as I said, why is it so complex? Well, we thought that because timing is important, maybe those sensory cues are helping males to investigate the sort of transient nature of there being other flies in the environment. So if we alter sensory cues, maybe we can alter the timing of this response. And that's exactly what we find. So if we remove the antennal segment, so we interfere with odor reception, then what we find is that males simply can't respond as quickly. So over here, we would expect that males should be responding after 24 hours with another rival, but they don't. They don't even significantly respond at 29 hours. They do eventually respond if you give them 3 days to respond. And they maintain their response for longer. So again, we've interfered with odor reception, and they are maintaining their response for after the rival has been removed for 12 hours, that was expected, but also for 24 hours, and that was not expected. So at that point, we should see that males are not responding anymore, but they actually are. And so this receipt of sensory cues seems to be important in that timing of their plastic response. We also thought that maybe this multiple sensory cues were helping to reduce off-target responses. So obviously males need to know things about the other fly in their environment. So as we've talked about the timing, they need to know that the male is going to be around for long enough to be a problem. They need to know that that other fly is male, and they also need to know if it's going to be a stern competitive threat, if another female is going to mate with it. So if that male is of the same species. And so we investigated the response of melanogaster focal males to males of other species. So we used other species that are differently related to melanogaster. So here, stimulans is the most related and virilis the least related. And when we don't do any sensory manipulations, then what we found was that melanogaster will make a sort of intermediate response to stimulans and pseudo-obscura, but won't respond to Yacuba or virilis. So a sort of intermediate kind of response, but it doesn't actually fit the pattern of relatedness to that other species. And then we removed different sensory modalities, and I know this table is a bit complicated, but basically we're doing similar manipulations, auditory, so sound, touch, and smell. And when we do that, what we see is where there are purple boxes, that's where we see an increase in the response to the other species. And these sort of orangey boxes are where there's a decrease in response to the other species compared to no manipulations. And so when we manipulate these sensory cues, we increase these off-target responses largely, so responses to heterospecific rivals. So again, it seems that although we couldn't identify that there was one particular cue that was really key in terms of species identity, we can see that if we do interfere with this, then males get it wrong more often. So we now know quite a lot about the inputs, we know about the timing of that response, we know about the combination of sensory cues that are needed, and we know about the effects of those sensory cues in terms of the speed of the response and in terms of being accurate. And then we started to investigate, because we could see that timing was important. Obviously, males seem to be learning and potentially memorizing their environment. And this is something, again, where melanogaster has been really important in understanding learning and memory mechanisms. So this is a sort of cartoon of the different types of learning and memory that flies exhibit. So we have short-term memory, medium-term memory, anesthesia-resistant memory, and long-term memory. And this is sort of hours of acquisition. So you can see that long-term memory for a fly is essentially anything over sort of 5 hours could be considered long-term memory. So in terms of our response to arrival, if males continue to respond for 12 hours, then this could be perhaps involving long-term memory. But certainly would expect that there must be some acquisition stage, so we should have some effects sort of earlier on that we can identify in terms of the genes involved. So these are genes that are involved in different types of memory. So this is rutabaga, amnesiac, and dunce, which are involved in short-term memory, and radish involved in anesthesia-resistant memory. So we probed these genes and which pathways are necessary. and this is what we found. So here we're using classic mutants. Over here we're using a knockdown, but basically a similar approach to interfering with the genes. Here, dunce doesn't seem to be important. If we knock out dunce, then males can still respond. But if we knock out amnesiac or rutabaga, then males are unable to respond. So those are necessary to the response. And over here, we looked at the effect of radish. So radish is a gene that is an exemplar of anesthesia-resistant memory. And at any point that we knocked out radish, males could still respond. So we don't think anesthesia-resistant memory is necessary. And actually, we did do an anesthesia treatment, so basically putting males on ice just before they mate. In fact, males that were anaesthetized couldn't respond afterwards. And so we know that we're not looking at this anesthesia-resistant memory, we're probably looking at this long-term memory. So we also investigated a particular part of the brain that might be involved in this. And this is the mushroom body. So this is the part of the brain that is involved in learning and memory associated with odours. And obviously we know that odour is potentially important in this response. So this is a fly face and it's looking out of the screen at you. And we've sort of coloured in a cartoon of the mushroom bodies. And so the mushroom bodies are made up of different lobes. These alpha, alpha prime, beta, beta prime, and gamma lobes. And what we can do is to interfere with transmission within each of those lobes of the mushroom body. And this is what happens when we do that. So this is our standard control. Obviously, they're responding. Here, we find that if we reduce neuronal transmission across the mushroom bodies, males can't respond. So we know that mushroom bodies are necessary. Here, if we just knock down in the alpha and beta lobes, then the flies are still able to respond. So that isn't the part of the mushroom body that's necessary. But in fact, it's this gamma lobe that is particularly important. And that is interesting because that type of learning and memory that is associated with this gamma lobe of the mushroom body is a type that is both long-term but able to be rewritten. So we think this is really important for the flies. This really matches what they're doing behaviourally. They are able to remember for some time that they have been with a rival, but the key to plasticity is that they can overwrite that memory. They don't keep responding if they've just once seen a rival throughout their life. They can rewrite that memory and so reduce their investment in the mating if there are no rivals around anymore. And so we think maybe this is an example of what is known as tailor-made memory, essentially using a particular pathway or type of memory that fits with the type of behavioural response that an individual needs to make to their environment. So we know something about the neurogenetic pathways involved in this response, and that the mushroom body is important. So sort of to sidestep from thinking about the mechanisms, as we were developing this work, we thought that this really seems like it's a lot for a fly with a little brain to do, that they are have this really sophisticated response to their social environment, and that maybe it takes quite a lot of cognitive reasoning to understand that there is another male around, it's of the right species, and it's around for the right amount of time to be a competitive rival. And so we thought that perhaps this type of social interaction might be involved in cognitive processes in the fly more generally. And that's not a new idea. So lots of people have been thinking about how social interactions, for example, affect brain evolution. But in particular, this type of work usually comes from mammals and usually comes out of the idea that there's a positive relationship between complex social bonds, in particular pair bonding, and brain size. And that actually this is found more in monogamous species, and in polygamous species, actually, there's a negative relationship. And so this suggestion is that really sexual interactions are, or sexually selected interactions, the competitive type of interactions, are really not important for this driving changes in cognition. But we thought that actually maybe this really wasn't the case. And perhaps looking at not mammals, we might be able to see that sexual competition really is cognitively challenging and could be involved in the evolution of cognitive processes. So we thought we could use our sort of fruit fly model to investigate this. So we think that reacting to this sperm competition environment is cognitively challenging. And so exposure to a rival male will stimulate learning and memory. But the problem is, how do we distinguish that it's a particular property of this sexual competition and not just social stimulation? So basically, having anything in the environment might stimulate a cognitive challenge and might stimulate these processes. And so we use our system to be able to disentangle these by using arrival of a different species. So obviously I said earlier that melanogaster males don't respond to virilis males, so this is a virilis male, in the same way. So they don't, or we suggest that they don't perceive them as sperm competition rivals. So here we can basically control for social interactions whilst also giving this challenge of sperm competition environment. And so this is our prediction. So if it is that sperm competition is particularly important, then we should see that a melanogaster male with a male of his own species shows changes in cognition, but not when he's with a male of another species. And if it's just that any type of social contact is stimulating, then a fly on his own will be worse than a fly with either a male of his own species or a different species. And so we set the males up in these different types of social environments, and then we gave them different tests of mainly learning rather than memory. But we gave them these tests. So we used a test we call Find the Virgin. It's not our test, other people have used it. But this is where males are given a set of females, one of which is a virgin female. And of course, that virgin female, one is more willing to mate with him, but two is also, in terms of sperm competition, is of higher quality, because obviously he won't immediately be in competition if that female hasn't already mated. And so males can learn to direct their courtship towards virgin females. So in this, he has to identify which that female is and then direct his courtship towards that, and they get progressively better to doing that over the task. In this task, we used visual learning assay, where the males are put in a sort of grid of heated spaces, and one square, this blue square, is cool, and so that's where they will aim to sit. And we will give them a visual cue, so this star, and we test whether or not they will orientate towards that visual cue when we change where that cool spot is. So whether they are actually not just sort of wandering around trying to find the cool place, but if they can actually orientate towards that visual cue. And then we also use this classic associative learning test in a T-maze where flies can learn to link an odour with a punishment. The punishment wasn't very severe, they just got shaken a bit, but they don't like it. And in basically all of these tests, males that were kept with arrival of their own species performed better. So not only were they better at learning, but they also showed differences in gene expression. So these are just a few genes that are associated with synaptic growth or basically increased neuronal growth. And you can see here that when a male is kept with another male of his own species, then we see increased gene expression of FUTCH and neurexin-1, but that doesn't happen when that male is kept with a rival of a different species. So we can see changes in terms of cognition, and we can see changes in terms of gene expression as well. And so we think we've been able to show that it really is this sperm competition environment that is cognitively stimulating for males. And actually we did all these tests on females as well. And females don't really seem to respond to females of their own species, but seem to respond a bit more to females of a different species. So a sort of sidestep there, but an interesting sort of link with perhaps general evolution of cognitive ability. So there we were kind of digging into what happens in the brain in particular, but we wanted to know across the whole fly what's happening in terms of gene expression changes. And so we used whole mRNA, RNA-seq to look across the whole fly, but we did split the flies into head and thorax and their abdomen so that we could try to capture different parts of the response. So in the head and thorax, perhaps gene expression changes that are to do with either assessing their environment or producing that behavioural change. And in the abdomen, any changes in the testes or accessory glands that might be to do with their change in ejaculate. And so we did this across a time course of males with arrival, so 2 hours, 26 hours or 50 hours with arrival. And obviously we're expecting that by 26 hours, males will have a significant response to arrival male. And what we found was fairly subtle but fairly consistent changes in gene regulation, both in the head and thorax and in the abdomen. So I'm just basically showing you here that we have initially a lot of upregulation in terms of genes when arrival is added, and those are to do with odorant sensing and touch sensing and that sort of thing. And we can see also some downregulation, and this happens quite quickly, so 2 hours after exposure to arrival. By 6 hours, we're seeing more downregulation in terms of there being arrival around. And then at 50 hours, we sort of see this sort of levelling off. Basically, the gene expression changes have sort of stopped. And so, as I said, lots of these genes do seem to be to do with sensing the environment, also producing the response. So actually, we did this experiment before we did the one about the previous one, where I showed you that FUTCH and neurexin changed in expression, and so we thought we might see expression changes there. And we also saw genes that have probably nothing to do with actually producing a response, but are to do with either stress responses, immune regulation, or indeed lifespan. So we can see lots of gene expression changes happening. So we see rapid transcription changes, but as I said, it's quite subtle, and by 50 hours it seems to be sort of shut off. But then we wanted to know how this is sort of controlled. And so we started to think about the role of the epigenome. So these are marks on the DNA or histones or chromatin that are essentially controlling gene expression. And a few years ago, a model was proposed where different types of plasticity have some involvement of the epigenome. So in terms of different types of plasticity or types of behavioural patterns, we could either have this type of pattern where there is some developmental stage, and then individuals will take one of two routes in terms of their behavioural patterns. Or we have this type of what the authors term plasticity, where there could be sequential plasticity. So again, there's a developmental stage, and then there's one behavioural type followed by another behavioural type. Or there's this labile plasticity, and this is the one that we've really been investigating, where individuals after development can flip between two behavioural sort of states. And in this model, they suggested that actually the epigenome was important for these two types of plasticity, but not for this very rapidly flexible plasticity that we've been investigating. Now, we thought this might not be accurate because in our transcriptome study that I just showed you, some of the genes that we saw changing are histone modifiers. So these are all histone modifiers which change in expression after exposure to a rival. So we investigated the role or potential role of the epigenome, firstly by a very kind of broad brush approach. So we used chemical inhibitors of epigenetic modifiers. So these are different inhibitors, and they inhibit these type of modifiers. So histone acetyltransferases, histone deacetylases, or histone methyltransferases. And in particular, these SAHA and sodium butyrate, which alter histone deacetylases, they seem to remove the ability to respond. So we think those are important. And also from the previous slide, RPD3 was a gene that we knew changed, and it is a histone deacetylase. And also SIR2, which is a similar gene and also a histone deacetylase. And so after this kind of really broad brush approach, we then used RNAI to identify whether RPD3 and SIR2 are important. And so here's RPD3. So if we knock down RPD3 ubiquitously across the body, then flies cannot respond to a rival. And we try to then identify the tissue that is important. And so we knock down RPD3 in the central nervous system. And this is a picture of RPD3 expression in the fly brain. So again, it's not my picture, but again, this is a fly brain looking out of the screen towards you. And RPD3 is basically expressed across the brain. And again, if we knock down RPD3 in the central nervous system, then it abolishes this extended mating duration behavior, which was, I guess, kind of expected. RPD3 has also been shown to affect other types of behavior. So in terms of ants, it is associated with behavioural changes between castes. And so RPD3 is clearly important here in this social response. The picture with SIR2, though, was a little bit more complicated. So when we knock down SIR2 across the body, indeed, we remove the ability of flies to respond. But when we knocked it down in the brain, in the central nervous system, flies are still able to respond, which again caused us a little bit of worry. This is a picture. This is James's picture. So again, the fly brain is looking out towards you. And here, blue is just the DAPI staining and green is the SIR2 staining. And you can see in the brain, there's really not a lot of staining there. So SIR2 is not really expressed in the fly brain, but James then went hunting for where SIR2 is expressed, and this is a fly testes, and you can see that SIR2 is expressed across the fly testes. And when we knocked down SIR2 in the testes, we removed the ability of the flies to respond in terms of extended mating behaviour. So somehow this effect is happening where essentially, well, flies are sort of syncing with their testes, and this work is in preparation. We haven't published it yet, but I really can't wait to write the media for that, and hopefully some people are going to pick up on that story. But weirdly, although SIR2 knockdown in the testes does affect the behaviour, it doesn't affect the ejaculate response. So we can still see that these males, although they're not showing the behaviour response, they do show an increase in offspring output. And so there's some feedback that's happening here where an effect in the testes is not affecting the offspring output, but it is affecting the behaviour. So hopefully I've convinced you that we've been able to really unpick with the fruit fly these different parts of this very flexible plasticity. And so we've been able to understand why flies respond to rival males in this way. It has really big effects on their fitness and also makes them smarter. We know about the type of inputs that are required and how important timing is for the fly. And we also know something about the mechanisms involved in terms of the neuro and genetic and epigenetic mechanisms. And I think, you know, we still have some way to go to really understand how flies are really able to do this, and in particular, how this kind of connection between tissues is coordinated so that the fly can essentially coordinate this response. I hope I've convinced you that flies really are interested in their social environment, and that there are lots of different types of responses that they can make, and these are interlinked. So we've seen a reproductive response that is also linked to a cognitive response and is also linked to aging and stress responses as well. And other work in my lab, we have also looked at responses in terms of their immunity, and we have a current project on their microbiome as well. So I'm going to stop there and answer some of the questions that I kept seeing pop up, but they come up so quickly I couldn't answer them at the time. But also just to thank all of the people and our funders for enabling this work. Thank you.
Thank you, Amanda. That was a fascinating talk. Very bright spot, right? From behavior to neuronal circuits to genetics. Very nice. As you can see, there have been a large number of questions. So let's try and take them one by one. I don't know how many we will get through. Wow, that's a lot of questions. Okay. First question is a very broad question. I was wondering if we know the extent to which the social environment affects Drosophila out in the wild. Is this being used as a model system to look at social effects largely because of the other information and genetic tools available for the species? And this is not with regard to studies on mating, but just sociality.
I think it's difficult with something like a fruit fly in the wild. Obviously, they're great in the lab, but in the wild, it is difficult to obviously track individuals and track how they're interacting with each other. Obviously, people who do work on wild species, can use things like our RFID tags to identify individuals coming into contact with each other and form social networks. But a lot of this work is based on lab observation. But lab observation in terms of trying to simulate sort of semi-natural environments. So other people, although ours are absolutely not simulating natural environments, other people do experiments where they try to understand social networks by giving flies a bigger sort of arena, giving them different food sources, and so trying to see a more natural kind of set of interactions. And certainly flies, so their propensity to form groups has a genetic basis, for example, and this type of response can be driven just by how you place the food in the environment. So a recent paper shows that just either grouping or spreading out food can prompt this or increase this type of response, because the flies are aggregating more around the food and so come into contact more and show this response more.
Okay. That was a question from Vidya. Here is a question from Amitabh Joshi. Are there effects on male mating duration or ejaculate after being held together with female flies without being permitted to mate?
Right. Yes. So the response to females seems to be a little bit more doesn't seem to be as consistent. So some people have found, for example, that males alter their investment in terms of behaviour and in terms of ejaculate, depending on properties of females, like whether the females are mated or how quickly they see other females. So whether it's a large group of females or a small group of females, it just doesn't seem to be as consistent as this response to males. So I don't know whether that suggests that simply having other females around will benefit males. Whereas the key thing is, are they going to be in competition? So I think maybe that is what's suggested here. In terms of not allowing them to mate, I mean, some of this is difficult to measure if you don't allow them to mate, but certainly there was a really good paper about essentially flies becoming a bit depressed if they're not allowed to mate. So male flies, if they know that there are females are around but they can't mate with them, they go and drink more alcohol. So there certainly are effects of females on males, but it doesn't seem to be quite as consistent.
Okay. Here's a question from Angana. If a rival male is only transient and not a threat, shouldn't increased exposure time show increased response from males?
Yes, and that's exactly what we do see in terms of mating duration. So that linear response. So the longer they're exposed to a rival male, then the longer they subsequently will mate for. We haven't actually tested the ejaculate response in terms of exposure time, partly because it's just a lot more effort than their behaviour. But I suppose we would also expect that to show us a similar response. Yeah.
Amitabh asked, redundancy is pretty ubiquitous in biological systems. So is it terribly surprising that multiple cues are being used by males to assess their rivals? So redundancy does seem to be something that can confer, particularly in this type of plasticity, can confer robustness to that response. So yes. Were we expecting the complexity? No, because there were a few other experiments in other animals that showed that individual senses were required. So in water voles, for example, just the odour of another male prompts the response. So actually, although perhaps now at this point thinking about whether that complexity was surprising, maybe not, but in terms of what was already published, then it did surprise us a bit, because, as I said, in other species
just single cues are necessary. And to be honest, I haven't really thought about why that might be in other species. Perhaps it's something to do with a threshold response. So just having a very big cue in one sensory modality maybe gets them over the threshold, and that's what other people have measured. I'm not sure. But yeah, that's something interesting to think about.
Thank you, Amanda. Since morning, we have a lot of new fascinating talks. Morning, we had mitochondrial DNA, claims, etc. in Australia, and now we have your work. Really excellent talk with new questions, new type of modeling, etc. And now I have a very basic question. Many times we have done experiments to look into the incipient reproductive isolation, male choice, female choice, all these things, right? We age males and then put them to females. And we calculate the mating duration, or even before the mate, they spend some time with the female. After spending some time doing some act, dance, etc., then they will mate. Now the question is, we have not looked, as far as I am concerned, we have not looked into the time of keeping these males away from the female, and also when we isolate males, we keep them not single males, but in groups. But we have not realized that keeping them together will have an impact on mating duration. And with this, with your experiments, etc., I suspect we have to relook into our data about the mating duration, the time spent with the female, etc.
Yeah, I'm really sorry actually because I think this affects lots of experiments where people aren't even looking at mating behaviour, but actually, you know, they, as standard, keep males. You know, they collect live, they keep them in big groups, and then they measure whatever. And I think this is something that I mean, sometimes people don't even mention whether or not they keep them in groups, and actually I think this might have quite a big effect. In terms of not seeing the female beforehand, yeah, I think so. We at a certain point get a bit worried that we really focus on the first mating of these males because it's the easiest thing to do. I think the studies where we have had males where we've kept them for their entire lifetime and given them lots of different opportunities, and the study where we showed that they are flexible in their behaviour, I think suggests that it's not a special property of that first mating, you know, that although there must be some learning in terms of reproductive maturity when they see a female, at least they do certainly carry on in their response to the rival throughout their mating history. So hopefully that's not too much of a problem.
How long this influence of isolation and put them together with one male, two male, three males, how long this influence will be retained in the males?
That's interesting. In terms of the behaviour, we know that it's around 15 to 18 hours. What we don't know is in terms of all the other things that we've been measuring. So we know that this affects their microbiome, their immune gene expression, and their cognition. We don't know how long that effect lasts. So I think that would be really interesting to understand, particularly, for example, in that experiment on cognition, if that alters their senescence. So in terms of their cognitive senescence, does that interaction, that social interaction, really kind of stop them from aging so fast in cognitive terms?
Okay, thank you. The last comment, this is a comment. In your first part of the talk, very clear data on the males, put them outside and put them together and bring them to the females, and copulation duration, etc. It is excellent data. And the moment you switched over to transcriptome analysis and epigenome analysis, I'm not sure. Why should we do it when the data is so clear? Because this epigenome and transcriptome, it is a generic things that are happening within the body. It need not be correlated with the work we have done with the duration of copulation, etc.
So I think it's important to understand those mechanisms so that we can understand how these responses evolve and what is necessary for the response, and also what are the off target, the changes that are induced by social environment that aren't to do with that response, I think are really important to understand as well. So that's really why we've started with trying to understand those mechanisms at that kind of level.
That can be happen if you can knock off the transcriptomes, or if you can inhibit epigenomic changes. Can you change this behavior?
Yes. So that's exactly what we find. So we haven't. I mean, obviously there are lots of gene expression changes from the transcriptome data that we just haven't been able to assess yet by functional genetics. Obviously, I think with the fruit fly, that's the really important step. Obviously, with lots of systems now, we can get transcriptome analysis, and that tells you that genes are changing, but it doesn't tell you that they are actually involved. I think being able to do that next step, and of course that's becoming easier with lots of different techniques. With CRISPR, for example, we can do that in more than melanogaster, obviously, but it's so much easier with melanogaster that we really can start to understand, okay, we see gene expression change. Does that actually mean anything in terms of the phenotype that we're seeing?
Okay, thank you very much. An exciting talk. Very simple questions, but broad answers. Excellent.
Thank you.
Thank you.
Okay, here is one more question. What sort of population was used to establish this phenomena? Relatively outbred, reasonably inbred, very inbred?
So we largely work on the Dahomey population, which is a large lab population that was established in the 1970s, and that's mainly what we work on. However, we've done similar experiments with other fly lines, such as Canton. Other people in other labs have also used other fly lines, and we see the same responses. So, given that the flies have been in a situation where their social environment doesn't change very much, they're always in big caged populations, but probably showing less sort of dynamic social environments, and yet this is a consistent response. So yeah, we've seen it in different fly lines, we've seen it in different fly species as well. So it's not just melanogaster that makes this response. We've done work with pseudo-obscura in particular as well with my collaborator Tom Price in Liverpool. So I think this seems to be quite a widespread phenomenon across fruit fly species.
Okay. Next are two related questions. Is there any evidence of variation in this mating duration response to male exposure across populations? And Mubarak has addition to that, he says that a number of rivals have been shown to influence male reproductive investment in other populations, which you did not find in your study. There are some others who have found it.
So we haven't really fully investigated how different populations are responding. We have tried to investigate in terms of Tracy has some sex ratio selection lines, so ones that are female biased or male biased, and we thought we might see evolution of this response in those lines, and we don't really. So whether that means that we're not either able to pick up the variation that is there, or that actually it's such a hardwired kind of response that actually that it's not very evolutionarily labile. I'm not sure. I think yes, we have some way to go to work out. Maybe there are more. There are population differences that we just haven't established yet.
Amanda, if I may add, there are some studies that we managed to do in the lab, which do show that this response can actually evolve. So there seems to be some population variation. These were studies that were very similar to what you did, but the question that we asked was whether this plasticity itself can evolve, and it looks like it can, at least in certain populations. So just as a further answer to that question, that's about it. Using a virilous male as a control for social but not sexual interaction is not particularly helpful, because how their presence is perceived as a social individual is hard to say. So do you want to comment on that?
I mean, it is. What we do know is that they, so we did find that they altered, so the melanogaster males altered slightly their latency to mate if they'd been with a virilis male, which they sometimes do with a melanogaster male, but actually it's not very consistent. So we think that they are at least, and we see some other differences in behaviour as well. So in terms of the activity of the flies, so we think that they can perceive that virilis male is there, and these two species are in nature can be found in multi-species groups together. So I think what we did was the best that we could do, and pick the best option there. And as I say, I think it's also interesting that I didn't show you the data for females, but seeing that the females are having quite a different response. Again, not something that we expected. We didn't expect them necessarily to respond, but again, if it's just about social stimulation, then we would expect females to respond to other females of their own species more as well, and that's actually not what we found. So yeah, I appreciate that there are problems with that, but I think it tells us something interesting.
Okay, so Medini has a question. What would happen if the age groups of test males and rival males are different? Would there be a difference in the outcome?
If the what? Sorry.
If the age of your focal males and the rival males were different, would you expect differences in the outcome?
So I think Stuart Whitby has investigated this, and I don't think, in terms of the behavioural response, that he did find a big difference, but we find a difference in terms of the microbiome response actually, and the age of the rival male. So this fits with previous work that has shown that the effect on lifespan of having other males around depends on the age of those other males. So actually, males will live longer if they're with younger males rather than co-aged males, which is what we usually use. So I think there are definitely age effects, but not on this specific sperm competition response.
Along similar lines, here is another question from Tuba. Do you think the focal male response will vary if they are kept with virgin males versus mated males?
I suppose so. I'm just thinking now whether we have assessed that. Have you assessed that? And I don't know about it. So mostly they're virgin males that are the rival males. might we expect a stronger response? I suppose mated males might have behavioural differences. They might be maybe more aggressive, perhaps. So maybe that might alter that response.
Yeah. Second question, why do you particularly focus on virilis as a rival? Why not some other species?
We just picked it as the one that clearly, from our experiment where we had the 4 different species as rivals, that the male clearly melanic acid didn't respond to. So that's just why we picked that one.
Is there a reason to focus on mechanisms rather than investigating the evolutionary dynamics and implications of this male exposure-dependent mating duration manipulation?
I think there's room for both. I think both approaches tell us about how this response came to be and how widespread we might think it is, and also how it interacts with other phenotypes, I think is really interesting. So the idea that this response might be involved in the evolution of cognition, which is not our idea either. So there was a theory paper published about how this type of reaction might be involved in the ability to actually estimate quantity. So in terms of number of males that are around, and so I think yes, there's room for both.
Praveen has a question. Which cues in rival males remain longer in memory of the focal males? And how do males identify virgin females?
Oh, I'm afraid we haven't done the full experiment to answer that first one. So if anybody wants to do that, please go ahead. Because actually, yes, that would be really, really interesting to work out if any particular cue was remembered for longer. I don't know, I'm afraid. In terms of virgin females, so there are both odorant changes in virgins compared to mated females, so they smell different, they also behave differently as well. So, for example, mated females are often more reluctant to mate, and so I think the males are tuning into both those types of cues in order to work that out.
Okay. So here's a question from Komal. He says that, in some populations, at least, the number of rivals has been shown to affect reproductive investment, whereas in your populations that the homies, it's not the case. Why do you think that the number of rivals did not matter in your population? Basically asking why is there a population difference, do you think?
Oh, I guess we would have to think about the history of this population, and so maybe because of the way that they've been kept for the last 40 to 50 years, in, as I say, a situation where their social environment isn't changing very much. Maybe just that binary, is there a rival or no rival, is all that they're queuing into. In terms of other populations, yeah, I'm not sure. I think probably the answer lies somewhere in there.
Okay. So Faraz has a question. Is there any study that suggests that any sexually selected display of Drosophila is correlated with costly organs that enhance sensory cues?
Oh, that's interesting. Yes, in terms of this idea of the costs of plasticity and the ability to produce plasticity, that's a really interesting one. do I know of any study? I cannot think of one off the top of my head. So, I mean, we did do a little bit of work looking at development and different social environments, and so males do invest more in accessory glands, for example, if they're expecting competition, but we didn't see a size difference, for example. But I can't think of, you know, in terms of, I'm not even sure what we might measure. Where I don't know if, for example, the mushroom bodies are plastic in size. So that is a really interesting thing to think about, but I can't think off the top of my head that somebody has.
Okay. I guess we have come to the end of the long list of questions. Let me just check once. I think that's... Oh, Professor Ranganath, please go ahead.
Is that a question? I marked this question long back, but I had to think twice before I can say that. Can I say that this is a type of social entrainment on male behavior?
Sorry, it's a type of...
Is it a type of social entrainment on male behavior?
So is it... I mean, it certainly involves learning.
Yeah.
So is it innate? Is that... well, they seem to be able to do it immediately. So maybe,
Okay.
yeah.
Thank you.
Okay. I think we have answered pretty much all the questions. That was a really long question answer session, probably the longest we have had. Thank you, Amanda, for a wonderful talk and taking all the time to answer all the questions. We really appreciate it. Thank you so much.
Thank you.